Hello everyone, it's Kemji and we're back again with our pharmacology course. Today we're completing the pharmacokinetics part. We explained the absorption, we explained the distribution, we explained metabolism, and now it's time for the last part of the journey of the drug inside your body and how your body affects it, which is basically pharmacokinetics, which is excretion. This is our topic for today. All right, we're gonna give a quick recap regarding the excretion process. First of all, let's say that excretion does not mainly happen inside the kidneys. It happens in lots of places. Your exhalation inside your lungs could be called, is called an excretion, a form of excretion. The substances that get excreted through the bile is a form of excretion. So many forms of excretion inside your body, but the main part of excretion of substances or drug inside your body, inside our bodies, is the renal excretion inside the kidney. So that's why we're explaining it or making a recap on how excretion happens in the kidney. And then we're going to get to some little details regarding the pharmacology of some certain drugs and the filtration, reabsorption, and secretion process. So without further ado, let's get started. So this is an artery that has everything that your blood normally has, nutrients, water, waste, everything. That goes into this structure all is found inside the kidney, inside the nephron. That goes into something called the glomerulus. The glomerulus is found inside, it's found inside something called the Bowman's capsule. So the content of the blood that we just mentioned, goes through a process called filtration, which is the first process that we're going to have today. What happens here is filtration. Actually, inside Bowman's capsule, about 20% of the blood plasma gets filtrated in here. All right, so 20% of the blood gets filtrated inside Bowman's capsule to get into this tubule, all right, inside the kidney. So then it goes through a structure that is called, or abbreviated PCT as straight in here, and PCT is an abbreviation for proximal convoluted tubule. The proximal convoluted tubule is actually the first place for reabsorption, which is the second process after filtration. We filter the drug, we made it filtrate. The blood, after it gets filtrated into the Bowman's capsule, we no longer call it blood, actually we call it filtrate. The filtrate of the blood. Right, so the filtrate that we have that has gone through the proximal convoluted tubule, we call it convoluted because of the convolutions that you find in here. Inside the proximal convoluted tubule, another process starts, which is the reabsorption. Some of you might ask, why would we need reabsorption? Simply because we cannot filtrate all the contents of the blood, including water, nutrients, salts, ions, and just get them to be excreted in the urine. We would lose very, very great benefits from these substances. So filtration happens mainly to get rid of the things we don't need, to get rid of the waste, the metabolite products that we discussed in the metabolism of the drugs, for example, and return back the useful substances or the useful things that we need, like water, like some nutrients, like some ions, and all of this we're gonna recap on right now. So that's why reabsorption needs to happen, and it starts in the proximal convoluted tubule. Reabsorption, and actually the main things that get reabsorbed from the proximal convoluted tubule are nutrients like glucose and water, and they get reabsorbed. I don't need to say inside the vessels again. Actually, something that I forgot to mention, your blood vessels, the arterioles, they get inside the Bowman's capsule. 20% of the blood gets filtrated inside the Bowman's capsule, and the other 80% that do not get filtrated, they go here to form these blood vessels, these capillaries that are found surrounding the loop of Henle. This loop is called loop of Henle. or the nephron loop. All right? So reabsorption starts from the proximal convoluted tubule. It keeps on going in the descending loop. This loop of Renal consists of descending loop and ascending loop. This is the descending loop. And this is the ascending one. Okay? Water reabsorption keeps on happening here, here, and here. Okay? It doesn't keep on happening in the ascending loop, and it makes sense actually. Yeah, in the descending loop water would be easier to gets reabsorbed, but it doesn't get reabsorbed in the ascending loop. The ascending loop, some ions like sodium, for example, get reabsorbed also. Okay? So the reabsorption process happens excessively inside the loop of Renal too. And regarding the distal convoluted tubule, which is abbreviated as DCT, some water reabsorption could happen inside there actually, but it happens mainly when ADH gets activated. For example, when you're dehydrated, when there's dehydration and you need more water to be reabsorbed from your filtrate, from the filtered blood that gets inside the kidney from Bowman's capsule and all this cycle that we mentioned, the pituitary gland, which is a gland that is found inside your brain, gets activated to secrete a certain hormone called ADH. And ADH is an abbreviation for antidiuretic hormone. Diuretics means the drugs or the things. It could be hormones, for example, that prevent, or diuretics means something that increases the output of the urine. So antidiuretic means that it decreases the output of the urine. So why is it called antidiuretic hormone? Simply because it increases the reabsorption from the distal convoluted tubule. Reabsorption means more water gets reabsorbed. More water gets reabsorbed and not reaching the collecting duct that reaches the ureter, that reaches the urinary bladder, that gets your urine out, means that less urine volume will be present. So yeah, let's continue. The green. So when ADH gets activated from the pituitary gland, lots of water will get reabsorbed again inside the blood vessels. So this is still reabsorption, and there's another part which is called secretion. Secretion happens when the substances or the waste or the drug that we need to excrete gets, let's try that first, gets secreted from the blood vessels to the tubules to be sent into the collecting duct to get rid of it in the urine. So 3, we're going to talk about the secretion. And from here, we're going to start the part that regards us as students or whatever who want to study pharmacology or want to know the relevance of the pharmacological part regarding the renal excretion. All right, let's first start with the filtration part. Some of the factors that might affect the filtration part is proteins, or let's say protein-bound drugs. Some of the proteins that might be found inside your blood are albumin, which are really important for us. But when albumin increases, or actually you have a high percentage or a high concentration of a drug that is protein-bound. Protein-bounds mean that it binds to proteins. It will not get filtrated inside Bowman's capsule. Why? Because it's big. Because it, yeah, mainly because it's big. It cannot be filtrated for something, because actually it has a repelling charge also. You could say that. Repelling charge to that found in Bowman's capsule, because proteins do have charge normally. So protein-bound drugs will not get reabsorbed, or not get filtrated, I'm sorry, inside the Bowman's capsule, which means that will not get excreted properly. So this is something that you need actually to take care of the protein-bound drugs, to know how to excrete them properly, because protein-bound drugs, the drugs that bind to proteins, will not get filtered properly, and at the end of the cycle will not get excreted properly outside your body, which will lead to some harmful causes. This is regarding filtration and one of the factors that could affect the filtration process. So what about reabsorption? There's a really interesting thing regarding reabsorption, which is a concept that we could say is called ion trapping. The other concept that we're going to explain regarding the reabsorption part is something called ion trapping. What is ion trapping? For example, let's say that you overdosed or took a very large dose of aspirin or phenobarbital, for example, and both of them are weak acids. We mentioned that previously in the absorption videos, yes. But we're going to mention it in a more specific way right now, unrelated way. So weak acids in the formula of the equilibrium formula of weak acids is all right. So you took a very high dose of aspirin, for example, a really, really high dose, and you do not want to keep reabsorbing this thing anymore. You want to reabsorption, I mean reabsorbing it into your blood, because this is actually intoxicating you. So you want to get rid of it. You want to keep it more in the tubules of the nephrons. You don't want it to get reabsorbed back again into the blood vessels. So what do you do in here? We mentioned a lot of times that what is the substance that would get more easily reabsorbed inside the blood vessels? Is it the polar one or the non-polar one? The one with charge or the one without charge? Easily, and we've discussed that before, the one without charge is the one that's simply going to be reabsorbed easily to the blood vessels. Why? Because the cell membranes of the blood vessels consist of, for the 100th time, phospholipid layer, and the phospholipid layer contains a phosphate group, and phosphate group is negatively charged. So a charge with another charge will repel each other. That's why polar compounds do not get absorbed easily through the blood vessels, because of the cell membranes, all right? So you need the substance to be nonpolar to get reabsorbed easily to the blood vessels. So what you do now? What you need to do is simply to prevent it from getting reabsorbed and get it excreted by keeping it inside the tubules and not get reabsorbed, is increase this. Increase the nonpolar part so it doesn't get reabsorbed easily. So how would you do that? By simply following the Le Chatelet principles, or the principles of the equilibrium chemical equations. We simply need to shift the reaction here. So what do we need to do to shift the reaction more here? We need to decrease the protons. And decreasing the protons means that we need to add something alkaline, something basic. Because something basic will decrease the proton concentration or the acidic concentration inside of this weak acid. So it will not get reabsorbed easily. So we will add something like, for example, in this case, sodium bicarbonate. Sodium. All right, this is basic. So it's going to decrease the concentration of the protons inside the weak acids. So this will lead to shifting the reaction forward towards the nonpolar form. The nonpolar form will be the dominant one. So this means that very less of the aspirin will get reabsorbed inside the blood vessels and most of it will get excreted outside the blood. That's why when you have something that's overdose of aspirin and getting it intoxicated from aspirin, you give him sodium bicarbonate as a medication for this, all right? And the same concept applies for weak bases, okay? The very same concept applies for weak bases, and the same idea basically. So if you have a drug like, what's a weak base? Let's play the equation first. Okay, so we have like amphetamines, yes. So the equation we will have, okay, so we're comparing this with this. Same concept. Which one do you want to increase in order to not get reabsorbed to the blood vessels? Exactly, you want to increase this and decrease, oops. All right, easy, yeah. It's okay. And decrease that. How you're going to do that specifically? Simply by, just a moment to fix that. Simply by increasing the acid, because you want to shift the reaction here. So you want to increase this here in order for the reaction to be to seek equilibrium, so it will shift backward. All right. So you want to add something acidic like ammonia, which is a source of protons in this case, right? Ammonium chloride, for example. All right. So, yep. So that's the ion trapping case. We call it ion trapping because we're trapping, in the case of weak acids, we're trapping the A negative that we have wrote, which is the form that we need to keep in order to prevent the drug from getting reabsorbed to excreted. And here we're trapping this ion in order for the drug to not get reabsorbed inside the blood vessels, and we explain the mechanism of both cases. This is regarding the reabsorption and the things that you need to understand. A weak base that applies, that this example applies on are amphetamines, like methamphetamine, for example, and weak acids that this example applies for, like aspirins and phenobarbital. All right, and we've talked about filtration, talked about reabsorption, and there's a very small part left in here, which is regarding the secretion part. And the secretion part simply are the substances that get secreted from the blood vessels to the tubules in order to get excreted. So these substances are the substances that are, we would say here, let's write first here regarding secretion, yes. You won't write anything new actually here, but the fact that these substances are, in order for them to travel without using any energy, which is something really basic, they need to travel from high concentration to low concentration. And in order for them to do that, they need to be lipophilic. Why lipophilic? Because the concentration of the same concept that we repeat over and over again, the concentration of lipophilic drugs will be higher in the blood vessels than in the tubules of the kidney. So lipophilic drugs will travel more easily or get secreted inside the tubules more easily because they're traveling from a higher concentration to a lower concentration. So lipophilic drugs, they are transferring from a higher concentration to a low concentration. So this is regarding secretion. They'll get secreted more easily than the drugs that are hydrophilic. Hydrophilic drugs will need active transport by specific transporters that are also found inside the tubules actually, in order to get secreted from the blood vessels to the tubules, because they're traveling from a lower concentration to a higher concentration. Hydrophilic drugs will have a higher concentration inside the tubules, not inside the blood vessels, for the very same reason that's related to the cell membranes. Right, this is and everything that gets excreted gets through the collecting duct at the end to get excreted to be urine right through here, right? So this is basically the very, very, very important basics regarding pharmacokinetics. If you understood these, I think, 5 videos about pharmacokinetics, you'll be able to grasp the whole context and content of pharmacokinetics. And yeah, that's it. And we're going to go on completing the pharmacodynamics and then starting to explain the pharmacology of lots of drugs acting on different systems inside our bodies in the following videos of this course. I hope you like this video, and if you have any comments or feedback, please leave it in the comments. I'll be more than happy to read your feedbacks, and thank you for watching the video, and have a nice day.